Hi, this is Sean Murphy, SK Murphy. I'll be the MC for today's PATCA briefing on nanoparticles. PATCA is a professional and technical consultants association. Learn more about PATCA at patca.org. We're fortunate today to have a briefing by Dr. Giacomo Vacca. In 2010, Dr. Vacca founded Kinetic River, a company dedicated to the development of advanced optical tools for particle analysis. Kinetic River's customers include the NIH, Stanford, UC Davis, the University of Kansas, Sony, Sartorius, and Italy's National Research Council. Prior to founding Kinetic River, he worked at Abbott Labs, where he invented laser rastering, an innovative, high-throughput approach to flow cytometry. He received his Physics BA MA from Harvard University and his Applied Physics PhD from Stanford University, where he worked with Nobel Prize winner Bob Laughlin. He's been awarded more than 80 patents to date. Dr. Vacca.
Well, thank you, Sean, and thank you, Theresa, for hosting this and for having me. I haven't been to a PATCA meeting in very long time, and this is my first time speaking at it. So I appreciate the opportunity, and thank everybody else who's joined in to listen to this webinar, as well as the people who are gonna check the video out in offline mode. So today I'm talking about a subject which is kind of exploding in the last few years, nanoparticles, in particular biological nanoparticles, and I'll be explaining what they are and why they matter and what are we doing about them and with them. So I guess as a matter of logistics. If you guys have questions for me, feel free to put them in the chat, and Theresa and Sean will monitor it. Will feed the questions to me as as the talk goes. There will be another Q&A section at the end in case there's some time left over and more questions. So the talk title is Extremely Small and Incredibly Dim: the quest to catch nanoparticles responsible for everything. Everybody can see the slides, I take it? All good? Great. Thank you. All right, so I'll quick roadmap. I won't dwell on the speaker bio. Sean did that well enough for me. I'll talk about what these nanoparticles are, their significance, challenges. What challenges are we facing in terms of detection and analysis? What technologies are we using to overcome those challenges? Pros and cons of those technologies. And then I'll be doing a deeper dive in specifically flow cytometry technology that I've been involved in for several decades at this point, that is particularly well suited, as you'll see, for analysis of these kinds of particles. Then I'll say a few words about my company, Kinetic River, and then we'll have the final Q&A. The audio is all good? Okay, great. Thank you. All right, so as promised, I will gloss over this because superfluous at this point. All right, so what are, in particular, biological nanoparticles? So I think everybody would have at least an intuitive understanding of what nanoparticles are. They're particles that are smaller than a micron, as a very generic definition. But there's lots of kinds of nanoparticles that we're not going to be talking about here, specifically, small crystals, even quantum dots that Nobel Prize, I think last year was given out for. There's lots of nanoparticles that are interesting, and plastics, microparticles or nanoparticles. We're not going to be talking about those. We're going to be talking about nanoparticles that are either biological in origin or have a direct intended biological effect. And so that's kind of the scope of what I'll be mentioning here. And I should put a global disclaimer. As much as I try to be very detail-oriented and fact-based, there are some details that I may have missed. In fact, I'm almost certain there are some details that I've missed. And so don't take everything you hear or see today as gospel. There may be some inaccuracies that I'm not aware of. And certainly This is not a talk that is focused on the biology, although there's some biology in it. It's really focused on the technology, the physics, engineering, and so forth. So what are these biological nanoparticles? Well, they're essentially very small capsules, little balls, somewhere between 30 and 250 nanometers. And there's a distribution of these sizes. As it turns out, there's many more of the smaller ones than there are of the larger ones. And so as we push the boundary of the technology further, we actually are seeing more and more of these nanoparticles towards the smaller side. But they're not all made equal. Some in these size range or gamut seem to matter more than others. And actually, there's a lot of active investigation, including at our lab, in conjunction with collaborators at the universities, to try and figure out what size range or function these different EVs might have. They're generally consisting of a bilipid membrane, as you can see here in this quite elaborate artist impression. They have a cargo. They have interior. They have content. Could be some kind of nucleic acid, proteins, lipids, all of the above. And they have surface proteins. Surface proteins is typically how they tend to first interact with neighbors or with other cells. And eventually the cargo, I don't know what happened there. Eventually the cargo is released wherever it needs to be released, and that's how the function is ultimately performed. So these nanoparticles are, something is very, very jumpy, sorry about that, are excreted by cells. So they're blebbed out, typically by cells. They can be called microparticles, extracellular vesicles, I'll shorten that to EVs for convenience, or exosomes. They don't all mean the same things. EVs generally is more like a broader class. Exosomes is a subset of that. Microparticles are the larger of the EVs in that range. And there's also other categories that are propping up, like exomeres. They're even smaller than exosomes. We don't have to get into that. Suffice it to say that there's lots of different categories that are being investigated for function, for biological function, for properties that may lend them to one kind of application or another. In addition to biologically derived nanoparticles, or EVs, as I will generally call them, you also have artificially made or artificially coaxed nanoparticles, such as lipid nanoparticles, or LNPs, and virus-like particles, VLPs. And both of these are used in medicine to carry cargo to where you want it to go. And I'll give you some examples of that. But that's basically one way to encapsulate some kind of a payload. It could be a drug, it could be a vaccine, and deliver it to a target organ, a target kind of cell, right? Something in, whether it's a type of blood cell or some kind of a tissue cell. So increasingly, these nanoparticles are being used that way. So I kind of started talking about this. So what do they do? So the naturally occurring nanoparticles, the range of functions that are being discovered, actively discovered right now, about what EVs and other small nanoparticles do is enormous, right? They're mediators of intercellular communication, or one way. So there's other ways. There's just free floating antibodies. Cells produce antibodies in the lymphatic system. The immune system produce antibodies. They go around and they tell other cells to corral the troops and fight off the invader. So it's not the only intercellular communication method, but is one that was basically not recognized until very recently. Very recently, I mean the last few years. And so you can imagine there's tons of different kinds of cells in the body, and therefore there's lots of different kinds of EVs that mediate communication between. So that's a huge field in and of itself. That's the good. The bad is that it seems that EVs are implicated in some way in various kinds. In fact, I think all the kinds of cancer that people have looked at in any depth, in particular metastatic cancer. So perhaps there's a role where EVs are sent out as sentinels by the cancer cell to find fertile ground, for metastasis to occur. They're also implicated in inflammation. So things that are generally not good, or very, very much not good. So there's lots of functions, lots of things that these EVs and other nanoparticles do. And this is just the naturally occurring ones. As far as the LNPs and VLPs, as I said, they're used to deliver the therapeutics to the target, be it some kind of an organ or a cell type. They're used for both vaccines and treatment, and they've been used most notably for COVID-19. In fact, they blew upon the scene in a big way with COVID-19. The mRNA that was in the vaccine, there was the vaccine was actually carried into the body through LNPs. And malaria, human papillomavirus, hepatitis B, also trials for pancreatic cancer are using these. So there's increasingly a recognition that these particles can be corralled, can be coaxed, can be used for things beyond what the body had initially designed them for. So I think it should be pretty clear. I kind of touched upon this, why they matter, but let's go one step deeper. So if you think of the body, right, the various systems in the body, various organs, lots of biofluids. Okay, so biofluid could be blood, obviously, could be plasma, so blood minus the cells, could be urine, cerebral spinal fluid, lots of fluids in the body, all of which contain EVs of various origin and various kinds. And so people started thinking, well, okay, so we found EVs implicated in this and that and the other thing. You can see here, right? There's a very long list and growing. This is only from a couple of years ago. So can we use them as biomarkers? Can we use them as sentinels, as proxies to tell us what's going on in a part of the body that is difficult to access? It's not the skin, to be clear. For example, if you could get an early diagnosis, an early warning system for pancreatic cancer, you'd have a winner, because pancreatic cancer is possibly the most deadly, if not the most deadly, one of the most deadly cancers known to mankind, to humankind. And that's because by the time it shows up, most of the time it's already too late. It has already grown, has been sitting there and growing, and by the time it's discovered, it's already ready to metastasize or has metastasized, and the patient's prognosis is very poor. If you can have an early warning system based on ideally non-invasive methods, that would be great. Be great because it could tell you, oh, look, you may have a stage 2 pancreatic cancer instead of a stage 4. That opens up the door for chemo, for surgery, for radiation, right? All sorts of tools that, again, most of the time, it's too late for by the time it's discovered. So biomarkers for early detections, huge field of action right now. You can also think of EVs as possible therapeutic tools. So we talked about LNPs and VLPs as artificially made nanoparticles for delivering cargo. But in a similar vein, you can think of EVs that are harvested from the body and selected in some way as themselves therapeutic tools for a particular disease. So you may have an instance where you take some kind of a biofluid from the body, you select a kind of EV based on some function or size or combination thereof, and then you re-inject it in the body to produce a certain effect. And right now, one of our collaborators at George Mason University is doing exactly that. In fact, we're collaborating with them on a new SBIR program that we just got awarded to establish what size fraction of EVs are most reparative in certain model systems of injury and healing. So lots of things going on. So it should be clear by now why this matters, right? COVID-19, all sorts of cancers, inflammation, injury, acute injury, heart disease, right? So the list is very long and growing. Okay, so great. So they're interesting, they're very useful, they're everywhere. So, okay, how do we see them? How do we find them? Well, that's the first challenge. They're extremely small. And by extremely small, I mean super, super, super tiny. Okay, here is I think it's a Giemsa stain of blood. And so these brownish things are red blood cells. These two are two monocytes, which is a type of white blood cell, not the largest, kind of middling white blood cell. And they're about 15 microns. Red blood cells are typically about 4 to 5 microns. Well, that dot right there is an exosome, 50 nanometers. Okay, that's to scale. I didn't play any games here. So you can see the challenge, right? So by now, and for the last, many decades, blood analysis, blood counting, routine blood counting, it has become routine. You can go to a doctor, get your 7 ml of blood out of your arm, and usually less than a day later, you have your complete blood count, right? You know how many white blood cells you have, how many red blood cells you have, how many platelets, and all different kinds of those. Well, that's great. Well, if you want to do the same with exosomes or EVs in general, much harder, because the tools were just not designed for that. They just fly right under the radar. So another way to look at this, is with what's the relationship between light, which is how we usually get the information about these kinds of blood particles, blood cells. What's the relationship between light and exosomes or EVs? Well, you can see here, typical wavelength of light that you might use to interrogate these kinds of things, 500 nanometers. Typical size of these things, 50 nanometers. It's a factor of 10. Lord Rayleigh, William Strutt, one of my personal scientific heroes from ages past, he would have said, can't do it, right? Because he was a product of the 19th century. In the 19th century, conventional wisdom, including his, said, well, if it's smaller than the wavelength of light, you don't have a snowball's chance in hell of seeing it. Turns out he was wrong. And luckily for us, he was wrong. But it's interesting how that played out. It took many, many, many decades, in fact, more than a century, for him to be proven wrong. And the way he was proven wrong has, in fact, unleashed a whole bunch of different ways that people are using this previously unsuspected way of detecting things. But let me take a brief detour to say, well, every time there's a challenge, there's also the possibility of an opportunity, right? It's a classic, martial art move. You turn a disadvantage into an advantage. And so what's the disadvantage here? Well, they're so small. What's the potential advantage you could turn that into? Well, because they're so small, there's a ginormous number of them. They're like trillions, literally, if not quadrillions. Actually, we don't even know really, but there's huge numbers of them in all the body fluids. Just as an example, you might look at EVs per milliliter of, tens of billions, maybe even more than that. Whereas the typical concentration of white blood cells in per milliliter is, roughly a million, right? A million, 5 million, something like that. So you have a huge disparity, right? And again, these numbers are taken with a grain of salt. There are biofluids with far fewer cells in them, like cerebrospinal fluid shouldn't have any cells in it, for example. I'm just taking blood as an example. The point is that the statistics can work for you instead of against you. And so you can see here this scanning electron micrograph of a cell and a bunch of exosomes kind of blebbing out from it. And so if you see this, you see, okay, well, this is one corner, one small corner of one cell. Look how many exosomes are just being spewed out, right? Close to 100, right? Just by rough count. And so that's the math that is at play here. Every cell in principle, not every cell will, but every cell in principle could be spewing out hundreds, if not thousands of these things, continuously. And so that becomes a much better numbers game for you. So whereas circulating tumor cells kind of ran into a wall because it turned out, so it was a great idea, right? So the idea of circulating tumor cells, you have metastatic cancer, cells are circulating in the blood. Instead of like trying to find the cancer, let's get some blood, find the circulating tumor cell in it. Says, aha, you have a circulating tumor cell, or 10 or 100. That means you have metastatic cancer, or some kind of cancer is about to metastasize, so you should go, try to find this cancer and kill it. The problem is that you can have metastatic cancer or cancer, and the number of CTCs in a 7 ml tube of blood, it could be less than 1, right? Just in terms of numbers. You'd have to look at a pint of blood or a quart of blood to find that 1 or 2 or 5 that will tell you clinically, yeah, you have a cancer that could kill you. And so that's the wall that the CTC field ran into. And that's why companies like Grail, and now there's controversy as to exactly what happened with them and what's going on. But the idea was, let's look at something other than cells. They went after DNA, nucleic acids, and all that. That's fine. But now there's an opportunity to look at EVs, because there's so many more of them than there are cells. So if you can find an EV and you can link it to a cancer, it's like, aha. And now you're not looking at doing a dialysis on a patient just to find that one CTC. You can take a tube, as you do when you do your annual physical, and say, great, there it is. 10 EVs, 100 EVs, 1,000 EVs. Let's assay those and figure out what's going on. So the second part of the challenge, of course, is that EVs are incredibly dim. By dim, I mean when you try to elucidate them, interrogate them by light, they're hard to see. And that's because of two things. What are you looking for when you're illuminating a particle by light? You're generally looking for two things. You're looking for scattering, you're looking for fluorescence. Well, for scattering, you run into the first problem here. This is a plot, John Nolan, thank you, of the scattering, call it the scattering signal, right? The strength of the scattering signal as a function of the size of the particle. These are just guides for the eye, right? In a semi-log plot, I understand that a power curve isn't a straight line, but just don't beat me on that. In this regime, it's approximately linear. In this regime, it's approximately linear. All I'm saying is that we know from many decades of work, not just ours, just tons of people, that scattering scales roughly as the square of the radius or diameter, doesn't matter, for particles in the range of cells, right? Cells and beyond. We also know that scattering scales as radius to the 6th power. Sorry about that. Again, something trigger happy there. Radius as a scale, as a power, 6th power of radius or diameter when you go below about 250 nanometers. That's a cliff you rapidly fall off, right? You can see that if you have a system trigger, a threshold of detection, if you will, at this level, pretty quickly you can't see anything. And that's where most flow cytometers, for example, sit, is above this threshold here. Therefore, they can't see most of the EVs they're caring about. So that's a problem. All right, so second part of them being so small, therefore very hard to see by light scattering, is also they're very small and therefore they're incredibly dim and hard to see by fluorescence. Well, so why fluorescence? Well, that's typically how flow cytometry, in fact, most of microscopy is done. You take a cell like this nice big fat white blood cell, which is covered in antigens that tell a trained observer what the cell is and what it does. Could be a lymphocyte, could be granulocyte, could be some dendrocyte, could be anything, right? And the presence and nature and amount of antigens on its surface are identifiers. They're calling card, business card, whatever you want to call it. So people for decades and maybe even a century have develop labels, which are formed of an antibody, which lock and key matches to the antigen attached to a fluorophore, some kind of a luminous thing that emits light in a different band than the light that is excited with upon excitation. So this fluorescence allows massively multi-parallel, massively parallel multi-parametric interrogation of cell samples writ large, right? So you can run now 30, 40 color assays on a flow cytometer and look at all these different kinds of cells simultaneously. But that's because there's quite a few of these sites on a cell surface. And if you look back at, what we're looking at just a couple of slides ago, yeah, you're looking at a size difference of at least 100, some cases even 2 or 3 or 500. The number of sites on an EV surface is hugely smaller, right? It's like the square of the diameter smaller. So that's a problem. So what constitutes dim for a cell in the order of 100 sites or antigens is actually about as bright as you'll get with an EV. So that's clearly an issue. So to restate the problem, right? You have these vaccines and therapeutic nanoparticles, the LNPs and the VLPs, good. You have these biologically occurring particles, which could be bad or could be good, could be used as therapeutics, but they could also be used as biomarkers of bad things. All of these are too small and too dim to be seen effectively using current tools. So that's the challenge, right? The challenge is to create tools that are better, more sensitive, more user-friendly. Sure, some exist, but they're not easy to use. They're too expensive, too slow, blah, blah, blah. Just inadequate, okay? So better tools. Okay, so to go one level beyond that, what's the beyond better? What does better mean? Okay, well, better means, okay, we definitely want to be looking at single particle detection. You don't want to be looking at aggregates of particles or ensembles of particles. You don't want to be looking at averages. You want to be looking at single particles, because that's been the kingpin of flow cytometry, is being able to look at single cells. That's what has unlocked the hugely successful immunology explosion over the last many decades. You want to do the same thing for EVs. You want to have the requisite sensitivity, first of all, to be able to see small particles. As I mentioned, the further we look, the more we see. And you want to be able to resolve them from one another, because if you just see, a big blob of signal, that doesn't help you either. You want to be able to look at multiple parameters simultaneously. Simultaneously being the key word here, because if you have to circle it back to see it, like, good luck finding, the same EV named Frank, okay? So simultaneous multi-parameter phenotyping. You want to have high throughput, because, yeah, you can use as you'll see, a scanning electron microscope to see with incredible detail a small field of EVs on a microscope slide or some kind of preparation surface. But that doesn't give you any number, any statistics to be able to make some kind of inference, some kind of decision about what that means. It just tells you, okay, there's 10 particles between 20 and 50 nanometers and 5 particles between 50 and 200. Okay, what do I do with that? I need 50,000 particles, then I can do something with that. You also want to have some flexibility. You want to be able to measure different ranges of sizes, because biological samples are messy. They don't comply with your demands. They like to be dirty and full of junk. And so you want to be able to have a system that adapts and can be tailored to different ranges of detection. You want to have stability in your system. It has to be easy to use. Particularly, it has to not clog. And it might be funny for me to mention that, since, like, what do you mean clogging? These things are so small, they probably passing right through your eyeball without you even noticing. You'd be right about that. The clogging is not from the EVs. The clogging is from everything else. A messy sample from some kind of biological source, like plasma, urine, blood, right? We'll have a ton of stuff. Obviously cells, but except for plasma, where the cells have already been separated out. But you'll have aggregates, right? You have proteins and lipids, so all sorts of stuff sitting there. And so that's what can clog your system. So if your system can only look, right, at the tiniest things, but you throw a boulder at it and it breaks, that's a problem, because most samples will have boulders in them. Okay, so I'll try to speed up because we're only about halfway. Well, I guess we're not so far behind. All right, so what are some of the main technologies that people have concocted to figure out, to detect, to analyze these nanoparticles? Well, I'll kind of go in reverse order in the slides that follow, but let me start with flow cytometry. Flow cytometry is actually kind of a late arrival to the nanoparticle party. Maybe not a late arrival, but certainly other things that being used for longer in some ways, until people realized that, yeah, well, you could use a flow cytometer to do this with certain provisos. Then there's microscopy. There's the old Coulter principle making a comeback. It was never dead, but it was superseded in many ways by other technologies. Other kinds of microscopy, video microscopy, and electron microscopy. All right, so let's take them one by one, try to go quickly through these. Excuse me about my cough. Electron microscopy. Here in this micrograph, I believe this is a transmission electron micrograph, is a megavirus. And you'll see the capsid shell, the protein sticking out and all that. Electron microscopy is fantastic. Okay, don't get me wrong. It's beautiful. It's a gold standard. So if you want to know exactly what the absolute size is of something that small, you better go to an electron microscope. Could be scanning, could be transmission, different pros and cons. I won't get into that. Has the best in class sensitivity. If you want to find something that's 10 nanometers, 15 nanometers, there's nothing else that will do that right now. However, extremely slow, very expensive, bulky. See it here, it kind of takes up a whole bench. And will only tell you really size and structure. It will not tell you what kind of proteins, it can't tell you what protein that is or what protein that is. It will just tell you, it looks kind of like a protein. And that's about it. So is limited. Even though it's the best at one thing, it can only do that thing really well. So it's not really a player in routine analysis. It's only used for establishing ground truth when you have something else and you want to anchor with it. Okay, so what about nanoparticle tracking analysis, or NTA? Well, this is an interesting technology. So you can see here a simulation, but it's basically, this is how it works. You take a video of a bunch of particles and you track each individual particle, right? And you see how far it goes. That diffusion is governed by equations that a very smart man once figured out. Actually, several smart people once figured out. Einstein, Stokes, Mr. Brown himself, I guess. And this diffusion equation is related to the size of the particle, among other things. So by measuring this diffusion rate, you can tell how big the particle is. It has good sensitivity. It can give you absolute concentration because it has a chamber that things are flown into. So you know the dimension of the chamber, you know how many particles you're counting. You can back it up from there. But it's not the fastest of processes and is limited in the number of parameters that it can access. Main players here are Malvern, the original NTA in many ways, and Particle Metrics, which was a second to the game, but in some ways it has exceeded the performance of the first. Okay, then we go to impedance measurement. So for those who haven't been steeped in this for the last almost 30 years, like I have, this is how blood counting was first automated. You took blood and you passed it through a tiny orifice. one cell at a time. And this tiny orifice, which by the way, was invented by Wallace Coulter. His brother was working with him, so the credit goes to both. It's called very appropriately the Coulter principle. And this is like late 50s, early 60s. Okay, so it's been around for a long time. Literally took a foil from a cigarette pack, put a pinprick in it, then stuck it in an electrolyte, put some blood on one side, and watched an ohmmeter as the blood went from one side to the other. So it's a very simple concept in many ways, brilliant. This is what made it possible to have automated blood counters at first. Eventually, this was superseded in many ways, although not completely, by flow cytometry, which is using light instead of impedance to detect the passage of cells through a certain volume. But this principle, is still valid. Yeah, it doesn't break just because we stop using it. And so people are not using it for nanoparticles. So you see, as the particle occupies the orifice, it excludes ions from going through. And so it reduces momentarily the conductivity of that path, which means that this impedance experiences a spike. You can measure the spike and you can count the particle. It needs an electrolyte medium. Okay, that's not so hard to do. It has pretty good sensitivity, but the dynamic range is extremely small. That's because you essentially need an orifice which is just a little bit larger than the particle you're looking for. Because if you make it much larger, your sensitivity isn't going to be good because the relative dip or relative spike in impedance is small. Just think of the cross section of a small particle in a big orifice. And if you make it obviously too small, your particle is going to clog the passage. So that's the limitation, right? It's prone to clogging, and therefore Spectrodyne in this case we can offer 5 different cartridges that you can use to measure the 5 different size ranges. It was like, okay, but what if I don't know what size range is in my sample, which I thought was the whole point, right? If you know already what the size range is, why do you need this measurement? So, it's a bit like the dog chasing its own tail. And don't get me wrong, there are use cases, right? Don't want to slam it. They've done a good job, but it has limitations. All right, then we come to super resolution microscopy. I won't get into the details of this. They also got the Nobel Prize for this. Betzig, Hell, and Milner, if I'm pronouncing them correctly. They figured out that instead of getting more photons out of your field of view, if you reduce paradoxically the number of photons, you can actually get much better at seeing where they're coming from and actually identifying individual emitters, individual molecules responsible for the emission of that fluorescent photon. And by doing that, with a combination of techniques that I don't have time to explain, you can actually reduce your uncertainty in the localization of that emitter to the point where you go from photo A, which is traditional fluorescent microscopy, to photo B, which is super resolution microscopy. It literally is beating the diffraction limit. Take that Rayleigh, right? Take that Abbey and take that Spare, all different kinds of folks who came up with criteria for how closely you could distinguish things. Here you can distinguish things 20, 50 times more closely spaced than was thought possible by the illustrious Lord Rayleigh, and until really a couple of decades ago. So fantastic invention. Has good resolution in X and Y, as you can plainly see. Relatively poor resolution, this is 2 microns, by the way. Relatively poor resolution in Z, so the axial direction. So, okay, not altogether the best. It's also inherently slow because guess what? If you have to reduce the number of photons that you are admitting at any one time, you're going to multiply your analysis time because you still have to get the photons. You just have to wait longer for it because you can't get them all at the same time. And it's expensive, fairly expensive. Main player here is ONI. They make a nano imager. Nice, very beautiful concept, but again, limitations. Then we come to flow cytometry. This, of course, is what I know best because that's what I've been doing for the last 25 years, 20 to 25 years. And, well, it may not surprise you to find out that I think it has the best capabilities. I honestly try to I try to come up with, okay, what is it not good for in terms of EV counting or EV detection or EV analysis. I couldn't come up with something. I could come up with things like, well, it could be better, yeah, but I couldn't come up with things like, yeah, this is a clear drawback. I could only think of things that it is better at than the alternatives. That doesn't mean that there isn't something that could be even better than it. I'm just saying that I couldn't come up with anything obvious that was a con to flow cytometry as applied to analysis of EVs. So flow cytometry is analysis, single particle analysis in flow of a liquid suspension of particles. Particles are measured by fluorescence and or scattering. You have to funnel and corral the cells or particles into a single file, which people have learned how to do. It's not that hard anymore. I mean, it's still hard, but people know how. And then you count, you analyze, you collect signals, and you gate and you understand. Very high throughput, right? So these days you can do, for cells, you can easily go 10,000, 20,000, 30,000 events per second. So you get the numbers, so you get the statistics, so you get the relevance. You can do multi-parametric analysis. So you can do simultaneously fluorescence, multiple channels of fluorescence, multiple channels of scattering. And you have, as it turns out, this was unexpected. We showed this, and we were surprised by it, even ourselves. Excellent resolution. You'll see an example of that. Main players, NanoFCM, company in China, was the first to produce a cytometer specifically aimed at this market, at this application. Beckman Coulter, which is the 820-pound gorilla in the field, has been doing flow cytometry since the days of, guess what? Wallace Coulter. That's where the name comes from. But only recently came up with the Cytoflex Nano, which is their answer to NanoFCM, first of all. And ourselves, we've been, very much, completely small player up to now, at least, in the flow cytometry field, because we focused on technology development, we focused on niche applications, but now we've really focused on this as our main application for the next several years at least. And so we have a product, the Delaware, that you'll hear about. So how do these products kind of sit and compare to one another in terms of the relative performance? Well, there's many ways to slice this multiple dimensional space. I chose two. Actually, one of my collaborators and co-workers chose two, and turned out to be a meaningful choice. You can look at number of fluorescent channels, which tells you how multi-parametric your analysis can be, and you can look at resolution, meaning how closely spaced can two particles be before you can't tell them apart. Not like physically, but in terms of size, right? And it turns out ONI is surprisingly is down here because, yeah, it has a good resolution X and Y, but not so much in Z, blah, blah, blah. This is the impedance Coulter counter. This is the flow cytometer from China, and this is the NTA, one of them at least. This is the Beckman Coulter cytometer, and this is the kinetic curve of Delaware. Excuse me. So you'll see some drawbacks, right? And in many cases, it's clogging. Clogging, or the inability, if it's not clogging, it's the inability to look at a range of sizes simultaneously, right? Or a range of sizes, maybe not even simultaneously. You chunk it up into multiple stages, is still a problem. The Delaware is the instrument that we developed from the ground up as targeted specifically to address these problems and be better, frankly, for the community than the tools that I was just going over. So what does the Delaware offer? It offers a quality control for vaccine production and vaccine R&D, as well as drug delivery R&D and production, a more accurate tool for quality control, a more powerful tool for drug development. as well as being a more versatile tool for research in biomedicine, again, applications and roles of EVs and exosomes. Ultimately, we see this as paving the way for early disease diagnosis and prognosis. In fact, we already started moving in that direction. This is a quote from one of our early customers. They're very happy, right? Very happy because it's objectively better. Objectively better how? Well, we have innovative optics, fluidics, algorithms, as well as overall system design, all of which play a role in delivering the kind of performance that our customers and partners are looking for. So I won't go through the litany of numbers, but suffices to say that we work very hard to make sure that the system was meeting the needs of the field as they are emerging and as they are evolving, frankly, over time. One of the things I'll focus on just as an example is something that, again, surprised even us, right? The 6 nanometer resolution, which you can see here from this, okay, back up. So this is a collection of particles, comes from a kit. These are polystyrene nanoparticles from Exometry. It's a fantastic product. They're extremely monodispersed, right? They're very, very, very tightly distributed in size, which is what makes this measurement possible. If they were like all over the place, you'd see a big blob, a big smush, and you can make heads or tails of it. But because they're so monodispersed, we could assay them on our instrument and pick out, okay, this is the 100 nanometer, this is the 125 nanometer population. And these are all coming at the same time, okay? We're not just sending them in one at a time. They're all mixed, and we can tell them apart, right? Not only can we tell them apart, but if you zoom in on the 125 nanometer population, the spread of that population is about 6 nanometers. That's electron microscopy level, okay? That's how crazy this is. Like I said, it surprised even us because I almost didn't feel any right to be for it to be so good. But there you have it. Okay, extremely good resolution, which means that if you have a distribution of particles, you can now see peaks and valleys where you didn't see them before, which means that you can see subpopulations that you were completely blind to before. And that is increasingly important as we home in on, okay, what subclass of the EVs in this sample is actually responsible for repair or for metastasis or for inflammation, right? So as people start studying these finer points, it's like, okay, we have EVs, now what? This is important. So we apply this and benchmarked it 6 ways from Sunday. One of the ways that we benchmarked it is by looking at human cell-derived EVs. These are commercially available from a company in Estonia called Biomed. They're about 90 nanometers. These are not as monodispersed as the very pure, very tightly distributed particles from Exometry, because they're human origin, and you know how messy humans are. And so these EVs happen to be engineered to express a fluorescent protein on their surface, plus they have naturally occurring non-fluorescent proteins called tetraspanons that are useful in kind of figuring out what they are and categorizing them. So we looked at all of them, right? We looked at the scatter in multiple directions. We looked at the membrane. We stained the membrane to make sure we're looking at the EVs and not debris. We looked at the intrinsically expressed protein. We looked at the other proteins by tagging them with exogenous markers, and we can see clearly the signals in every case. And so this is actually a very good benchmark for us, for anybody. In fact, this is a very popular product because it allows you to kind of cross-validate different technologies. So I'm kind of barreling through the end here, but I did want to mention a couple of important things. So one of it is we started collaborating with Chris Ward, which is essentially one of the world's experts in polycystic kidney disease. Polycystic kidney disease is a terrible disease. It affects about 400,000 people in the US alone. And it's bad, right? It's bad because it's debilitating and ultimately can be fatal. Very painful, hard to treat. If you catch it early, not so hard, easier, right? And so it'd be good to know that you might be susceptible to this, or you might be developing polycystic kidney disease before it becomes, before your kidney becomes the size of a football. And so this biomarker that he was responsible for isolating actually looks at that. And this is a normal urine sample, but the same biomarker has different relationships to other biomarkers or other metrics in patients affected by PKD. And so we did the preliminary results, and now we are looking to expand this collaboration and really pave the way towards an early diagnostic using fully non-invasive methods, right? Urine. How easy can it be to just literally piss in a cup?
So just for clarity, so from a urine sample, you believe you've now able to detect a biomarker for the polycystic kidney disease. What is the current, absent this, what is the current method for detecting it? Is it you have to become symptomatic, or?
Yeah, so there's genetically you can find, because it's an autosomal dominant genetic disease, so you can be tested for that. The problem is there's many variants. I don't know the full biology of that, but Chris tells me that you can test, but there's so many variants that it's hard to pin it down. It's hard to know whether you actually have the bad variant or not so bad variant. So genetically, not the easiest thing to pre-screen for. So it's ultrasound, it's MRI, right? Or
But that's kidney size. When you say ultrasound, you're looking at kidney.
right, exactly.
Right.
Yeah,
So that's well into a symptomatic.
exactly.
Right.
Yeah.
So
And if you want to, if you can look at polycystin 1, which is one of the markers that Chris has identified and created, using Western blot. But in Western blot, you're looking at averages. You're looking at bulk. You're not looking at single particles. And that's where, you're taking Monet's water lilies, acetone, and then, shake it into a bucket. It's like, okay, what's in this bucket? Yeah, there's some blue, there's some green. That's basically what you're doing with Western blot. Instead of like, wow, that water lily is amazing. That's the difference, right? Is seeing the individual nanoparticles and their expression levels instead of dumping it all in the bucket and hoping that you see some kind of separation of the blue versus green.
so this feels very much like you've opened a window into a new world. I mean, you're now seeing you're able to monitor particles, count them, and assess them at a size which is like 1,000th of where we were before, if we're just looking at
Yes,
blood cells, right?
yes, that's correct. Yeah, so size determination is going to be important. We're actually working on doing that automatically so that the user can get a size histogram, which is kind of what they have grown to expect from other tools like NTA, like electron microscopy. So we're working on that, and we're well on our way to being able to release that. And that's about it. I just did want to say a couple of things, and we're almost done, and just want to slide it in. Kinetic River, just about the company generally. So we are based in Mountain View, so we're local to the PATCA team and many other groups, but obviously we can be reached by anywhere. And many of our customers do come from anywhere, anywhere in the country, in fact, anywhere in the world. We've had a lot of collaborators, partners, customers, clients from all over the place. We focus on flow cytometers and analysis of particles. Our strength is in optical instrumentation. We do technology consulting that goes very deep into that, but also outside of it. So we've done flame diagnostics, for example, as a project for a customer. We design, develop, and manufacture our own products. We have 26 issued patents with 5 pending. Very innovative company. This is some of the people on the team. You'll see our illustrious host, or one of them at least, on the team there. And Rosemary, who's now doesn't have the camera on, but we have a picture of her anyway. We've been very fortunate with our advisors, very fortunate with the team that we've been able to assemble. Super great, very hardworking, and very, very good at what they do. We also offer nanoparticle analysis services. So for customers who may not be sure or want to kind of get their feet wet first, they send us the samples, we analyze them, we send them a report, right? Rinse and repeat. And Chris Ward is one of the first customers to take advantage of that because it's a way for them to actually get the benefit of the Delaware before or while they're looking for other sources of funding to get their hands on a national instrument. And the last slide is I will mention that we are in a fundraising round at Kinetic River. We have been bootstrapping since we got started. And at this point, with Delaware in particular, we see an inflection point. We see the opportunity to actually grow and scale up into a market that is much bigger than what we have been doing before. And so I just wanted to put that out there in case any of you might be interested in following up or know somebody who might. And I will stop there.
So we'll extend for a minute or two. If folks have questions, this has been one of the most exciting talks I think we've had all year in terms of bona fide high technology. Any questions for Dr. Vaca before we break? Well, I appreciate everyone coming, and I always learn a lot when I go to one of your talks. You have a very, very skillful way of kind of connecting the dots and putting things together. We will be back next month with another PATCA briefing, and thanks everybody for coming.
Thanks very much for having me. I appreciate it.